Can you guys see that? Is that cool? Okay. Okay, so we're going to talk about cellular respiration, and it literally is the opposite to photosynthesis. It is the absolute opposite. Okay? So first of all, let's just start with the definition. It is the process, okay, by which sugars are broken down to make ATP. So the process by which sugars are broken down to make ATP. Now the goal is this, is to make a total of 38 ATP. That's the goal. Okay. This process occurs in plant and animal cells. So plants go through photosynthesis and make their own sugars, and then their cells break that sugar down to produce ATP so they have energy. Okay? Okay, so let's start with the equation. So C6H12O2 plus oxygen produces carbon dioxide and water. It is the exact opposite of photosynthesis. These guys are your reactants, and these guys are our products. Now, there's a lot of other stuff that is made in between to help make the ATP. Okay, so there'll be a few other molecules, but the main gist of it is glucose plus oxygen gas is going to produce carbon dioxide and water. Okay, there are 3 steps. The first step to cellular respiration is called glycolysis. Glycolysis. So glycolysis, this happens in the cytoplasm of the cell. Our second step is called the Krebs cycle. And this happens in the mitochondria, in the matrix. So, and sorry, I don't have a colored picture for you, but this kind of weird looking shape, this stuff in between all of these folds, that's the matrix. And it's kind of like a fluid-like substance like cytoplasm. So I'm going to write fluid-like substance. And the Krebs cycle happens here. So we're going to say matrix of mitochondria. And then our last step is called the ETC, or electron transport chain. You guys have already heard of this. The electron transport chain. And this occurs right here in the inner membrane. So I'm going to put inner membrane of mitochondria. So right here, so this kind of membrane that contains all of the matrix, that's where the electron transport chain occurs. This is where most of the ATP is made, is in this last step. We got a little bit made here, a little bit made here, but the bulk of the ATP is made in the electron transport chain. Okay, so with our time, I think we can knock out the first two steps, and then we'll just have to finish the third step on Friday. Okay, so flip this over. Okay, so this whole thing represents a cell. So I'm going to write a cell, and this whole thing is a cell. Okay, this circle inside, this represents the mitochondria. Okay, this stuff all around here is my cytoplasm. So we are going to start inside of the cytoplasm. Okay, we're going to start right over here. And this is step number 1. This is glycolysis. Okay, so in glycolysis, the major thing that happens is this is our first breakdown of our glucose. So we're going to do C6H12O6. This is our glucose. And this is going to break down into 2 molecules. We call them pyruvate molecules or pyruvic acid. And each of them has 3 carbon atoms in it. So this is what I'm going to do. I'm going to write C, C, C. So I know it has 3 carbon atoms. And I'm going to write pyruvate, P-Y-R-U-V-A-T-E. That's just the name of the molecule, P-Y-R-U-V-A-T-E. So glucose is going to break down and is going to make 2 pyruvate molecules in glycolysis. Okay, it's also going to make 2 ATP. Put this in red. These stars represent ATP. And I'm going to have you draw one more circle right here. And these circles represent a molecule called NADH. NADH. So it makes 2 ATP and 2 NADH. Now I'm going to come down here and I'm going to put a little star. Okay, these NADH molecules, okay, they are electron carriers. And let's see if I can make that bigger for you. There we go. Okay, so they are electron carriers and hydrogen donators. I don't know if I spelled that right. Doesn't look right to me. But. So their job is literally to take electrons and to take hydrogen to the last step. Okay, that's their job. Because in order to crank out all of the ATP that has to be made, we have to have hydrogen ions and we have to have electrons, okay? Hence the electron transport chain. So we have to have something to provide us with that. And those NADH molecules do that, okay? So that's what happens in glycolysis, okay? So now what we're going to do is I am going to move over into the mitochondria. And we are going to look at step number 2. So I'm going to put right here. And this is going to represent the Krebs cycle. Okay, so the first thing that happens is each of my pyruvate molecules are going to move into the mitochondria. They just shift over because we need it to break down even more, okay? So I'm going to put my CCC, and I'm going to write pyruvate, P-Y-R-U-V-A-T-E. So they're just going to shift over. And they're going to go into the mitochondrial matrix. They're going to break down more, okay? This is where one of our products is made, okay? We've got 2 products. We've got water, we've got carbon dioxide. This is where one of them is going to be made. Okay, so first of all, each pyruvate produces a molecule of ATP. And due to space, I didn't have enough room, okay? So each of these stop signs represent 3 carbon dioxide. 3 carbon dioxide. Kind of makes sense. If I got 3 carbon in each pyruvate, it can break down and make 3 carbon dioxide. So 3 carbon here, carbon dioxide here, 3 carbon dioxide here. We're not quite done, okay? Each pyruvate also makes a total of 4 NADH. So I'm going to put 2 NADH, 2 NADH. Again, more hydrogen donators and more electron carriers. So I'm going to do the same thing here. 2 NADH, 2 NADH. I got one more molecule that's going to help NADH out, okay? And that's represented with the triangle. So FADH2, FADH2. These guys have the exact same job that NADH has. They are electron carriers and hydrogen donators. They are going to be utilized in the last step, okay? So right now, of our 38 ATP, right now we have 2 made in glycolysis, 2 made in the Krebs cycle. So the rest of them are going to be made in the very last step, which is the electron transport chain, okay? We'll stop right there. We'll finish this tomorrow. If you're not going to be here tomorrow, I will videotape again